join me in welcoming to the Distinctive Voices podium, Dr. Clarence Peters.
Well, thank you for that introduction, and hello to those of you out in the audience who I know. It's always good to have somebody who can lie for you when they're introducing you, but nevertheless, I want to talk a little bit about Rift Valley fever and some of the issues, but I'd like to lay a little background in terms of emerging infectious diseases. Now, this may seem like old data, being, 175 to 50 million years ago, but in fact, I think it's important. When I first started working on emerging infections as an entity, I didn't realize that I'd been working on several of these viruses before they became emerging infections. We'll come back to that. When I began to work on emerging infections, I realized I wasn't going to be around to see what was going to happen to a lot of these infections. So I started looking at historical aspects as well as current aspects. You can see from this diagram that 50 million years ago, the continents had already formed. So there was plenty of time from then to now for an evolution in each distinct land area of its own distinctive flora and fauna, and these continents in turn are chopped up by rivers, mountains, and other natural divisions. So you can pluck several mammals out of Australia, and you'll find that, sorry, that's Antarctica, Australia's down here, out of Australia, and you'll find that they're marsupials, and you'll know immediately they came from Australia. Similarly, if you grab a few mammals from Madagascar and you end up with lemurs, you're going to know it's Madagascar. Well, the same is true for viruses. But since the 1500s, we've been moving back and forth between these continents, and we've been carrying with us the viruses, the arthropods that might transmit them if they're arthropod-borne viruses, and the potentially the vertebrates that could be amplifiers. Now, this is kind of but the current word that everybody uses is wonky. This is kind of a wonky slide, but I think it's very important that you understand the concepts, that there are different epidemiological strategies for each one of these viruses, and there are several that can be put together in terms of viruses that persist in humans. They either are chronic infections or more often latent infections. And to give you a good example of this, one that you're likely to be familiar with, is the herpes zoster virus. It causes chicken pox, goes away as far as you can tell, but it burrows into the dorsal root ganglia, and later, when your T cells are old and tired, as you might be, it'll pop out and recur as a varicella eruption, and in turn be available to infect other humans. So it doesn't need any sorts of intermediaries and so on. Another kind of epidemiological type is the viruses that don't persist in humans at all, but have to continually hop from human host to human host. And a lot of these viruses, the most notorious anyway, have been controlled by vaccines, such as smallpox, which has been eradicated except for a couple of laboratories, measles, polio, and so on. I just point out to you that when these viruses seem to be eradicated, like smallpox, or even, let's say, like polio, which is on the verge of eradication, at least some people hope so, that if those viruses are reintroduced, they'll find a naive population and they'll spread extensively if there's no way to control them, either through quarantine or through stored vaccines. And finally, the virus I'm going to talk about is a member of the zoonotic group. They're maintained in non-human hosts, and they only incidentally spread over into humans. And this slide gives you an example of what might happen for one of the viruses that is maintained solely by human-to-human transmission. This hunter-gatherer woman probably has herpes zoster or a related or a precursor of that virus in her dorsal root ganglia. And if she activates it, she can spread it to her offspring and maintain the continuity of existence of that virus. And those viruses have been with us for a very long time, and their phylogeny corresponds to human or ape or even rodent phylogeny. Just to continue that theme, here are some viruses that probably evolved only during urbanization. When we were hunter-gatherers, we weren't able to transmit these viruses from one to another. But the precursors from one animal or another have crossed over into humans and become viruses that we recognize as human infections. And of course, we know this from the historical records, but more importantly probably from isolated island populations and tribes that have been sampled before they have any extensive human contact. Returning to the geographic theme, and I think gives you a good feeling for what may be going on here. This is Asia, and this is Australasia. You can see New Guinea, and Australia itself will be right down here. And it shows several lines that have been named for their proponent. Probably the most important and most accepted one is Wallace's line. And one of the things that is very interesting about this is it splits between Bali and one of the Indonesian islands. These islands are close enough together that you can see from one island to the other. You can tell that the birds are different, the trees are different, the bushes are different. Somehow, for thousands, if not millions of years, there's been independent evolution on either side of this line. What's interesting, and you can't see, is that there's one set of viruses on the western side and another set of viruses on the eastern side. For example, chikungunya virus is found extensively on this side of the line. Closely related virus, but different, called Ross River virus, occurs on this side of the line. And when they cross over, for example, as Ross River virus did about 20 or 30 years ago, you get massive epidemics if receptive conditions exist. So we're kind of sitting here, and we have, in terms of, particularly in terms of the zoonotic viruses, bad things that are just waiting to happen. A very smart guy named Josh Lederberg organized a meeting at the National Academies, which you'll, of course, are familiar with. This was the National Academies in Washington. And they produced a monograph called Emerging Infections, Microbial Threats to Health in the U.S. But they really didn't mean just the U.S. They meant all over the world, because these things can be transmitted or transferred from one area to another. In 2003, the group met again, and I was fortunate enough to be part of this group. And we picked and picked at the conclusions of the first report, and we decided there was nothing that we wanted to change or thought needed to be changed. I would list for you all the different things that are recognized as contributing to emerging infections. But one of the things that I think is, rather than listing them, one of the things that is more important to understand is that they're changing exponentially. If this is air travel, number of people who cross continents annually, exponential increase. Disturbance of the environment, exponential increase. Things are changing more and more and faster, and I think we have to be very concerned about that. And one of the real concerns that I want to talk to you about is Rift Valley fever virus. Now, some of you may be thinking that these viruses that emerge and cause problems are actually mutations. And in fact, I don't believe personally that there's any one virus that exemplifies this except influenza virus. Most of the other viruses that have been involved in emerging infections are really occupying a new ecologic niche or are spread by travel and transport. Not to say that after a virus hops across a global boundary or hops into a new niche that it doesn't evolve to adapt itself to that niche. That does. I mean, these are very variable viruses, but the real drivers, I believe, of the opportunities for the viruses to evolve and for us to suffer is not in the intrinsic changes in the viruses. And here's an example that you may want to think about. It's taken us a while to evolve, and it doesn't necessarily take them all that long. But the question is, will they find a place where they can survive? Because some of these genome changes in the viruses are lethal to the virus or affect its fitness so that it can't really prosper in a new area. Probably the first emerging virus that we've dealt with in the U.S. is yellow fever virus. And this is from a gravestone in Philadelphia. And this just says that that this marker is for a 10-year-old victim who fell due to yellow fever in 1793, aged 10 years. You know, these viruses have been trying to get where they want to go for a long time. And in the 1700s, Philadelphia was a receptive area, and they came via the Caribbean to Philadelphia and caused a massive epidemic and ravaged the Gulf Coast and the Caribbean for a long period of time. We're increasingly recognizing that bats are important reservoirs of some of the worst viruses that we've come across. This happens to be a fruit bat who's hanging, I could say upside down. That would be upside down for me, but right side up for him. And when he manages to infect horses, because we're disturbing the environment in Australia, where the first outbreak of this type occurred, that these animals become more and more of a problem as they roost in palm trees and feed in other areas. A closely related virus to the horse virus is currently active in Bangladesh. And it's active because, well, in California, you appreciate the importance of wine. Well, in Bangladesh, they appreciate the importance of palm wine. So they have containers in palm trees, and they harvest the liquid in these containers periodically through the tap they put into the palm. And if one of these guys has been drinking at the same site or pollinating the palm trees, he can deposit the virus in the material that they're collecting and result in encephalitis. The other thing that's interesting to me, and I think would be interesting to you about the viruses that are coming out of these flying foxes or fruit bats, is that they're paramyxoviruses. Now, paramyxovirus doesn't have to mean anything to you, but it's a kind of virus that's very commonly infecting humans and so on. But the paramyxoviruses in question, when they're looked at from a phylogenetic point of view, are a completely new branch on the family tree of paramyxoviruses. So it's an example, I think, of the fact that there's a lot of things out there that we don't know about. Another thing that's important is that a majority of these viruses have come from Africa. And I think there are two reasons. One is that we don't know very much about Africa. And the other is that Africa is a tropical area and therefore capable of supporting many different kinds of vertebrates and arthropods. And this is just an example of why we don't know that much about them. This is a picture of the Lassa fever research station that CDC maintained in Kenema, taken in 1979. It doesn't look that much better today, but you can see these two buildings are not exactly the kind of buildings you would expect to find on the campus here at Irvine, or even in a primitive place like Texas. I mean, it's they're not. There's not a lot of surveillance going on in Africa, and this is an African hospital. This happens to be an Ebola patient, and you can see the person who's actually taking care of the patient is fairly well protected. Gloves, gown, a mask with positive pressure air blowing over the mask. But in fact, what you can't see is that tonight this guy's wife will come to help take care of him because all the nurses and doctors will be gone and nobody's feeding him. So they'll come to bring him food, and if his wife is tired, she may sleep in the same bed with him. So there are real differences in what we see and don't see in Africa. Now this is a photograph of an area of Africa that's called the Great Rift Valley, and it runs from Tanzania up through Kenya and across to the Red Sea. And if you think that the Rift Valley is an obscure place, it's not. I mean, it's an area that produces a lot of coffee. I don't think you have to worry about these beans at Starbucks, but it's still something to think about, that many of the places where we get things that are important to us are places where these viruses originate. Now, the virus called Rift Valley fever virus was first isolated in the Rift Valley, as you would suppose. They had a massive epidemic among European breeds of sheep and cattle, and they also had febrile disease in humans. Now, I'm sure some of you in the audience know more about this than I do, but from what I've been able to ascertain from the literature, sheep and cattle were actually brought to Africa and distributed 2,000 or 3,000 years ago. And they developed a certain amount of hereditary resistance to the infection by the virus. I don't mean immunity in the sense of they had antibodies, T cells, and so on, but I mean resistance in the sense of early innate resistance. When the Brits came to this area and introduced breeds of sheep and cattle that had a better productivity for their purposes, it changed the equation. I might as well admit now, we have no idea what the original reservoir for Rift Valley fever virus was in terms of amplifying the virus. But these, I'll also point out that the colonial powers at this time were very, people think about the colonial powers as interrupting African types of governance and African customs and relationships and so on. But really, that was a byproduct. The real product was they wanted places to exploit in terms of minerals and bringing their crops to grow in African soil. If you look at the French colonies, for example, they had a completely separate administration that sent scientists to Africa to explore their colonies, to make vegetation maps, soil maps, to collect biological specimens, and to take care of the people who were going there to make the colonies profitable. And these maps exist today. You can find them, and the Brits did pretty much the same thing, but less systematically. Well, when this epidemic occurred, there were a couple of veterinarians from the UK who isolated a virus from these sick sheep primarily. The virus was originally identified as a filterable agent, which at that time was equivalent to what we now recognize as a virus. And they used not fancy cell cultures, electron microscopy, et cetera, but this virus killed mice very readily. So they knew they had a virus. It was filterable. They put it back into sheep. It caused the disease in sheep. Fine. And then they looked around, and there was one of the docs that had been brought over to take care of the settlers, and he inoculated this into a few humans and reproduced the human disease, which was shows that we have progressed, because this wouldn't happen again today. At least I hope not. And they were able to take sheep up to high altitudes where there were no mosquitoes and show that transmission stopped, and thus in a period of a few months, they determined most of what we know about Rift Valley fever virus. We know the distribution is fairly wide, but the actual epidemics that have occurred are relatively small in number, and I would bring to your attention two of these. One was in 1974 in the Republic of South Africa. They had just had a case of Marburg virus that killed the index case and one of the two nursing sisters and made two other people sick. And so they were very alert to hemorrhagic fever, because the cause of death with Marburg virus is generally hemorrhagic fever. So when Rift Valley fever came through, they were expecting an acute febrile illness that was non-fatal. So all people who came to the hospital and they had fever, myalgia, malaise, were dragging around, they said, go home, take 2 aspirin if you can afford it, and come back in the morning. But there were a few cases that presented with hemorrhagic fever. They were hospitalized, and virus isolation studies were undertaken, and they showed that this virus, in a small number of cases, probably 1 in 100, could cause hemorrhagic fever. Another important epidemic was the one in Egypt, which showed that the sub-Saharan distribution that had been seen previously could be interrupted by introduction of the virus to a distant area. And there was an enormous outbreak in Egypt, which resulted in numerous cases of hemorrhagic fever, over 600 reported deaths. And the other outbreak I would call to your attention is an East African outbreak that occurred in Kenya and other East African countries, as well as the Horn of Africa. And it was related to El Nino changes in climate, and it was a huge outbreak. And it smoldered along until it was spread actually to the Arabian Peninsula, and I'll show you that on the map in a second. This is a map taken from a NOAA satellite that demonstrates the greenness of sub-Saharan Africa. This is an artificial color map, and red is really green, indicating high rainfall, and blue is fairly high rainfall, and this, of course, is an arid area which is not was not thought to be very important for rift circulation. This is a close-up picture of the virus we've been talking about. C shows aggregation of nucleic capsid and incorporation of membrane-bound glycoprotein to produce particles, and the mature particle we see here with these peplimers around the outside, which are the glycoproteins of the virus. And in the center of this slice, you see nucleic capsid gathered together, and you have the complete particle. We have a better picture of it now. This is cryo-electron microscopy. I show this to tell you two things. One is the way we measure the number of viruses in any sample, sheep blood, human blood, mosquitoes, whatever. We inoculate a monolayer of cells, in this case virocells. We wait a certain period of time, and then we apply neutral red, and we find that the neutral red stains the live cells, and it doesn't stain areas where all the cells are dead. And we call these plaques, and you can see that there are numerous plaques, but what's more important is that the size is different. And that tells us that RNA viruses in general, and this virus in particular, are very variable in their genetic constitution. So in one small amount of a virus preparation that's applied to this plate, you can see there's already an extensive change in the genetics of the virus. If you pick one of these little bitty plaques from a Pasteur pipette, you can grow it out, and the first time you grow it out, no problem. It's all small plaque, and it's attenuated for mice. But if you try to take this any further, it reverts, and you'll find mixed plaques again, and it'll be virulent for mice. So this is an example, I think, of how viruses have a built-in adaptability that only requires the proper selection, the proper atmosphere for them to grow out. This is just a laundry list of the susceptible animals and what happens to them, and the bottom line is that the less innate immunity they have, the higher the virus titers are, and the more likely they are to die. And if they're very young animals, they will probably die almost uniformly. If they get any kind of viremia, the virus will invade the fetus and you'll have abortions. And this has tremendous implications for the economy of a country, because not only will they lose a lot of animals, but people don't want to import animals from countries that have this virus circulating. In fact, it circulates all the time, but it takes an epidemic to wake up some of the countries that are potential recipients. Also, if you're a herdsman and your animals are aborting, you lose this year's lamb crop and next year's harvest of animals. These are some infections we did at USAMRIID, and this is a ewe who was immunized with a vaccine that you'll hear a little bit more about while she was pregnant. The vaccine didn't cause abortion of the animals. As soon as they were born, they suckled and took colostrum, and they themselves became immune, not through the vaccine, but through maternal transmission of immunity. And this is a lamb that came from one of the control sheep, and you can see the lamb is virtually necrotic. The placenta is largely destroyed. So vaccines are one way to deal with this, but there are a lot of problems with vaccines, and maybe we can talk about that later. Why am I talking about Rift Valley fever virus, aside from the fact that it's one of my favorite viruses? And that's because, there are many different mosquitoes that can be infected by this virus from reasonable concentrations of blood in a blood meal that they may take. We know that almost any place, based on observations in Africa, where there are vertebrates that will develop a nice viremia to infect more mosquitoes, you can have epidemics. So we have areas with a lot of sheep and cattle. We know that these areas of Kenya and Tanzania where rift occurs in epidemic form periodically and in endemic form every year, these are prime game areas so that a lot of tourists go there. We've documented Canadian and Dutch tourists that have returned with rift infection. We also know that it's a bad virus for humans. You can get bitten by an infected mosquito, or you can work with blood or generate an aerosol in the laboratory and infect yourself very easily. And of course, we know that it's been able to go into new territory. Just wanted to emphasize that the actual material itself is not a problem for butchers. The people who get infected are the people who butcher the animals, but not in the butcher shop. This is a snapshot that was taken in Egypt in 1977. You'll notice a little bump here. This is a camel carcass. This is an area where camels are commonly eaten. And I happen to have brought with me something that I ripped out of the Wall Street Journal this morning. And they apparently gave the president of France a camel. This illustrates the fact that you shouldn't assume that your customs are theirs, and also that there are just a lot of different attitudes. They gave him this camel, and he left it with a family in Timbuktu to take care of while he was away, and they butchered him and ate him, the camel, that is. And the Mali officials said, "We are ashamed of what happened to the camel. It was a present and did not deserve that fate. As soon as we heard of this, we quickly replaced it with a bigger and better looking camel. The new camel will be sent to Paris." I can hardly wait to hear what happens when the camel gets off its 747 or whatever in France. We could talk a minute about human disease. Relatively short viremia. Antibody occurs and terminates the viremia and eradicates the virus. Probably 98% of humans have a brief febrile disease. It's very debilitating, but has no serious outcome. But some humans will develop hemorrhagic fever, and most of those die. Some humans will develop retinal vasculitis, probably about 5%, and this results in lesions that diminish vision or can lead to complete blindness, and a few develop encephalitis. This is a woman in Egypt with hemorrhagic fever. You can see, well, 2 or 3 things. One, that she's pregnant, but that's not necessary to develop hemorrhagic fever. You can see these little hemorrhages in the skin, which are actually raised and indicate a vasculitis. And you can see that she has areas where intravenous lines or blood was drawn have big spreading hematomas. These considerations led to an argument between the physicians and the veterinarians. The veterinarians saw mostly sheep and lambs with overwhelming disease, and in fact they call this disease a hepatitis. We got a couple of samples from an outbreak in Kenya, and Sri Saki stained them. And one of the patients had very extensive liver disease, as manifest by the stain that shows the viral antigen in red, and that goes with the veterinarian's opinion. But another of the patients who had hemorrhagic fever clinically had only small focal involvement. And that's what led us to really believe that this was a true hemorrhagic fever and not a hepatitis. This is I guess I could have done a little better job in terms of workmanship on this slide, but it tells the story. A rhesus monkey is inoculated to provide a model for human disease, and you can see there is a very rapid rise in viremia, but it's terminated in 3 or 4 days by the emergence of antibody that's capable of neutralizing the virus in vitro. And this is part of the extensive evidence that we have that antibody is what's important in this disease. This shows the interferon response. And as most of you probably know, interferon is a naturally occurring compound that makes cells resistant to viral infection. This is a monkey that developed hemorrhagic fever. But this is the slide that I wanted to show you, and that is, it's not the amount of interferon or the amount of virus that's important. It's how soon the interferon response occurs. This is a number of hours post-infection when interferon was measured. If their first interferon response was in the first 6 hours, the animals didn't even get sick. They didn't miss a biscuit or lay down in the bottom of the cage or anything. Although other animals that took up to 12 hours for their first response were sick, up to 24 hours. Some were sick, but a few died. And if you didn't make your interferon response until 30 hours, you were going to get sick and die. I think this is just an example of how some of the innate factors that have nothing to do with antibody, T cells, and so on can be extremely important, and interferon, of course, is one of those. If we look at the interferon response, and I think this is kind of a grisly picture, but if we look at the encephalitis issue, this was taken in an Egyptian fever hospital, and this 5-year-old child died with encephalitis. And this goes to part of the problems with why we don't know very much about Rift Valley fever. First of all, everybody here wasn't dumb. I was taking the picture. This guy's an experienced pathologist from NAMRU. This guy is dean of Uniformed Services University, and this guy is first assistant to Tony Fauci today. But we were all sitting around, and we couldn't really get into anything until we located a Coptic family that allowed this autopsy to occur, because Muslims do not allow autopsies in general. This child had encephalitis. This is one of, as far as I'm aware, 3 autopsies in patients that have died from encephalitis. So there's very little information available on Rift because of where it occurs and the infrastructure involved. This shows one of the ocular lesions looking in with an ophthalmoscope through the cornea and back at the retina. This is the optic nerve, and this is an area of vasculitis with a lot of white exudate. To the extent this resolves, you're going to get your vision back, and if it doesn't, you're not, and here's a little hemorrhage over here. We don't understand the pathogenesis of this lesion at all. Now, what about the natural history of rift? Well, there are areas which in Kenya are called dambos, which are actually circular, and you can see that around the edge of one of these areas, there are grasses and sedges, and in the center, still, even though this is dry season, there's a little mud there. If you notice this poplar tree, this is rainy season. You see the same poplar tree, but this dambo has flooded. And the way rift maintains its continuity, at least today, is by being transmitted transovarically by a particular species of Aedes mosquitoes into the mosquitoes come in oviposit in these areas, and the eggs lay there perhaps as long as 10 years documented. And then when it rains and floods, it doesn't just take a little rain, it takes a real flood. They will initiate their hatching program, and the newborn mosquitoes will come out, and they will take a blood meal, and this vertebrate host, if they take the right vertebrate host, they will result in the initiation of further transmission, because if the rains are that hard and that consistent, there will be a lot of mosquito vectors around, and they will be able to hatch out newly infected vectors, and they'll be able to infect humans either through mosquito bite or through contact with aborted fetuses and so on. So this is a pretty nasty disease, all in all. One advantage that we have over the virus is that it requires a lot of rainfall to hatch, and this helps us, for its eggs to hatch, this helps us to surveil what's going on. If there's just rain, you'll maintain vegetation growth, but if there's more rain and you have the standing water in the dambos that I showed you, then these mosquitoes will hatch, and there will also be an opportunity for other species of mosquitoes to hatch in quantities. So you can get secondary transmission, primary transmission from transovarial, and then later transmission from a variety of other mosquitoes. And of course, if you get a lot of rain, you'll get generalized flooding. Well, in 1997, we had a very interesting occurrence. These are NOAA satellite images of East Africa and the Horn of Africa in a non-epidemic year. These have been adjusted to what's called the normalized vegetation index, and they are, the green in this case is a false color that represents a balance of two particular wavelengths on the receptor in the satellite. And you can see here that compared to 96 and 97, it's a lot greener and a lot greener, indicating increased rainfall, and there was a humongous rift epidemic that year. This is an example of what happened to people who were living out in the flatlands of Kenya. And these are temporary structures that have been put up by people who are ordinarily nomads, but their area of distribution and movement has been shrunk by the flooding. They're crowded one against another. They're absolutely inundated with mosquitoes and losing lots of animals. The roads are all blocked so that the little surveillance that could be accomplished was done by helicopter. That epidemic stopped itself, but what's important about it is it was one of the biggest rift epidemics we've ever seen. And it also, just to say, it involved Egypt, Sudan. There was a lot of involvement in all the way down into Somalia and Tanzania. And that was what set the stage for rift across the narrow Red Sea. Animals are routinely brought across, sheep are routinely brought across because the Saudi folks and the Yemenites like freshly slaughtered sheep. I don't know why, we can go into that, but they like the variety of sheep that are widely grown along this area. They're called Somali fat-tailed sheep, and they are brought across, they're slaughtered, and they're more or less barbecued. But somebody forgot to look at the Rift Valley fever situation, and if you'll notice, you probably think of the Arabian Peninsula as being a big sand pile, but in fact, on this side of the Red Sea, remember, this is a continuation of the Rift Valley. So this is low compared to the elevated areas on either side. And you have rainfall along this particular littoral area, and you get a lot of mosquitoes. And there was a massive epidemic in Yemen and Saudi Arabia. In fact, this occurred around the time of the Hajj. And the Saudis don't like to talk about it, but there were virus isolates made right outside Mecca. So this was a tremendous risk in terms of further extension of the virus into additional areas. We also made a live attenuated vaccine against Rift Valley fever virus, and this has been sort of dragging in terms of its development, mainly for financial reasons. But this is just a diagram of Rift Valley fever virus. It has 3 segments, one small, medium, large. Virologists are imaginative about some things, but not everything. So that's why these are known as small, medium, and large, and not, But anyway, and these little stars show the mutations from the parent virus. The virus has been in about 60 humans, and it's quite attenuated. The only side effect we found so far is headaches. These are 19 people that we did 3 or 4 years ago, and you can see, just look at the red indications. There's not enough virus to readily measure quantitatively, but we could isolate virus by serial passage, blind passage, and we sequenced it and looked at the mutations, and there are no reversions. The little purple dots indicate the mutations compared to parent virus, and these guys indicate the mutations that were found compared to the vaccine virus. And you'll notice there are no reversions, and there are relatively few new mutations. And we could put this back into immature host and show that it maintained its attenuated phenotype. We could also use this to test in sheep. This was done in pregnant ewes, and you can see the antibody titers were quite high, and you can see that the vaccine was able to stimulate appreciable antibody titers. So we have, in effect, a vaccine for the virus, both to use in humans and to use in domestic animals. And that's why I entitled this talk, do you call the doctor or do you call the veterinarian? We don't know yet whether you can stop human disease by vaccinating domestic animals. We just don't know whether humans in isolation with a plentiful mosquito environment can be protected by vaccinating domestic animals and stopping the amplification of the virus there, because humans develop high viremias when they're infected, and they could infect mosquitoes. So I think this is a Francis Bacon quote that really applies to emerging infections, because if we sit around, there'll be movements of vectors, reservoirs, viruses, and we'll have to deal with them in one way or another, or we can get out ahead and deal with them ahead of time, which is what I am hoping that we can do with Rift Valley fever virus if financial situations are right and there's enough political will to move forward on this. Thanks for your attention.